The following content is provided under a Creative Commons license. Your support will help MIT OpenCourseware continue to offer high-quality educational resources for free. To make a donation or view additional materials from hundreds of MIT courses, visit MIT OpenCourseware at ocw.mit.edu.
We're going to get started, and today we're going to move forward with the protein degradation module and looking at the degradation chamber of E. coli, ClpXP. So this is a degradation machine. And effectively, what we see in this case, and as mentioned in lecture last time, is that there are gigantic chambers that isolate protease active sites. And so we're going to examine this particular machinery as a paradigm here. So the ClipP system was first identified in E. coli, and it's highly conserved. And what we'll see is that there's encapsulation of an active site in a large degradation chamber. So there's two components, ClipX and ClipP. And so we're going to look at both of these components individually and then see how this machine works. And so ClipP this is a proteasome. Okay, and it's a serine protease. So we talked about serine proteases last time. So there's the catalytic triad of serine, histidine, and aspartate. So there's formation of a covalent acyl enzyme intermediate. We learned that the serine residue is the active site nucleophile. And what we're going to see is that this degradation chamber has 14 active sites. Okay, and what this does is degrade proteins into short polypeptides, so into peptides of about 7 to 8 amino acids. Okay. And so if we consider a cartoon of the structure, what we find is that we have two back-to-back rings here. Okay, and each of these rings is a sevenmer, so we have two heptamers. Okay, in terms of size, approximately 90 angstroms here, and approximately 90 angstroms across. Okay, and then this region between the two rings is sometimes referred to as the equator. Okay. So here we have two back-to-back rings. Okay, and these rings generate a chamber. Okay, and proteins upwards of about 70 kilodaltons can fit. And so if we take this looking at from the side and just rotate 90 degrees to ask what does it look like from the top. Okay, so this is a side view. Okay, what we see is that there's a very small pore here. Okay, so we have the 7 subunits, and then in the center, there's an axial pore. Okay, and this pore is small, about 10 angstroms in diameter. Okay, so in thinking about that size, we need to think about the size of some large protein, right? If we have something on the order of 70 kilodaltons with a fold, that protein's not going to fit through this hole in that state here, okay? So it's too small for a big folded protein. Okay, but then basically if we take this and rather than just looking at the top, we cut through and ask what's going on in the interior, what do we see? Okay, so cut through. What we see now is that there's a chamber of about 51 angstroms. Okay, so this is the interior degradation chamber. Okay, so one question we're going to address as we move forward is how is it that a polypeptide gets through this hole into the degradation chamber that can accommodate, a protein up to 70 kilodaltons? Okay, so small axial pore versus large degradation chamber here. Okay, so we'll look at some structures of ClipP and then go on to ClipX. So what we're looking at here are effectively what I've drawn out in cartoon form on the board. So here we have the side view of ClipP. We have the top ring, the bottom ring, and here's the region between the two, the equator. If we look at the top, here they're describing the axial pore as a portal. Okay. And here's the cutaway view, so this hole is very small. And if we look at the cutaway view, what we see is the degradation chamber here. Okay, and basically the 7 different serine protease active sites are shown here. Okay, and this is a side view cutting through the middle here. So we see that these serine protease active sites are down in this region here. If we take another view and look at, again, this is cutaway, so cut through the side view, what we can look at here is the machinery in the active site. So we learned last time about the catalytic triad with the aspartate, histidine, and serine. And in this particular structure, there's a serine protease inhibitor bound to the serine side chain here. Okay, so that conserved machinery. If we look at the structure of an individual ClipP subunit, those are shown here from several different organisms. What we see, if we can think about this as the top part, and this is the bottom part of one ring, we see there's a region with axial loops, there's a head domain, and what's called a handle region. Okay? And the catalytic triad is located at the juncture of the head and handle region. And you can, again, look back to the cutaway views to orient that within the whole chamber. Okay, these axial loops we'll see are important for interaction with ClipX, and we'll talk about that component of the machine in a moment. Here, again, just structures of ClipP from various organisms, E. coli, Streptococcus, human. We see that they're all very similar here. So what is ClipX? Moving forward. Okay, so ClipX is effectively an accessory protein. Okay, and in some respects, we can think about it as a lid to the proteasome. Okay, it's a hexamer. Okay, so there's a mismatch here in terms of the number of subunits in ClipP and ClipX. This is different from what we saw with GrowEL, GrowES, where they are both heptamers. ClipX is a hexamer, and it's an AAA, so AAA plus unfoldase. Okay, it's an ATPase here. Okay, and effectively what we'll see is that ClipX has an important role as an accessory protein that unfolds the polypeptide that's destined for degradation by ClipP. So it unfolds the polypeptide, and we're going to have to ask how as we go through. Okay, and in addition to unfolding, it also threads that polypeptide that's being unfolded in through the axial pores such that it can reach the degradation chamber. Okay, and threads it into the degradation chamber. Okay. And so if we look at ClipX from a top view, again, we have a hole. And we have a 6-mer, okay, here. So if we take a look in this particular depiction, what we're seeing are the top views and the side views. So here we have ClipP, here we have ClipX. And what we want to ask is, how is it that ClipX binds to ClipP? And how is this mismatch in terms of the number of subunits accommodated, right? So it's not that one subunit is precisely going to act with 1 because we have 6 in throughout, because we have 6 in ClipX and 7 in ClipP. And so if we think about how ClipX binds to ClipP, what we see is that we have the 6-mer, so we're looking from the side view. Okay, and there's some loops that are called IGF loops. Okay, so these are tripeptide motifs, and they're flexible. Okay, and these loops interact with hydrophobic regions of ClipP. Okay, and this flexibility helps accommodate the 6 versus 7 subunits. So if we look here, here we see these tripeptide loops, okay, and see we're only seeing 3, but there's 1 per subunit, so 1, 2, 3, 4, 5, 6 here. And then what's shown here in red are the hydrophobic regions of ClipP where these can bind, okay. And where do we see these regions on ClipP here? They're a bit removed from the axial pore here, okay. So how many of these loops are needed? Just to note, there's been studies done where these residues are deleted, and the question is how many of these motifs are important for this protein-protein interaction. And what's been found in test tube studies is that a minimum of 2 are required to get interaction between ClipX and ClipP. Here. Yeah?
Is it known how many actually interact in vivo? Like, do all 6 interact at any given time?
I would presume so, but I don't know, right? So we very much think of this as coming together, like as shown here, there. But don't know, Joanne, do you know? No. I would say it needs to be pretty stable. Like, there's always dynamics, but as we see how this machine works, right, this hole is going to have to allow the polypeptide to thread through and get to that through that axial pore for the polypeptide to get in the degradation chamber. So you'd imagine you want that to be lined up quite well in order for it to be efficient there. Okay, so what are these AAA plus ATPases? This is a very important group of ATPases. So what the name means, ATPases associated with various cellular activities. And they're super duper given this. Hopefully everyone gets a AAA plus on the exam tonight. The superfamily is involved in many cellular functions, and take a look. So many diverse functions. So membrane fusion, trafficking of vesicles, cytoskeleton regulation, transport, organelle biogenesis, DNA replication, transcription regulation. And what we're really interested in here is protein degradation. So they come up in a variety of processes. And although these processes are very different, all of these AAA plus ATPases share a common protein architecture. And I'll just point out that there's an ATP binding module, and some details are given here in terms of the motifs. And then really what we'll focus on in terms of aspects of this course is that they form oligomers that are ring or cylinder shaped, and they're all hexamers here. Okay. And so of importance to ClpXP, these ATPases have the ability to remodel conformation of macromolecules. And so here we're focused on unfolding, yeah.
This might be a really silly question, but what are ATPases that aren't associated with cellular activities?
Well,
is this definition based on the architecture?
Strictly, yeah, I mean, there's...
If you look at tRNA synthetases, right? They have ATPase activity. Hundreds of proteins have ATPase activities.
Right, so...
They hydrolyze ATP to ADP and PI.
Right, but that's a cellular activity, right?
So like what aren't AA plus ATPases?
Well, your amino-acyl tRNA synthetases are not AAA plus ATPases. What we'll see in terms of the non-ribosomal peptide synthetases, they're not these AAA plus ATPases, right? So these, I mean, yes, okay, all ATPases, the enzymes in a cell, is going to have some role in a cellular activity, right? So maybe this name isn't very helpful, but what's common about all of these is that they share this common structural motif. They form these hexamers, but within that, there's quite a bit of diversity because they have all these different functions, so we can just see that here to some degree. Oops, and there's a typo, which I'll fix before posting, but if we take a look just at two examples here of different hexameric rings, so these are two different AAA plus ATPases, what do we see? So in common, they're both hexamers. In common, they both have an axial pore here. But we see different elements of secondary structure. And granted, these are both depicted in a bit of a different way, but if we look here, I mean, look, we have these alpha helical regions around the exterior that we don't see here, okay? And in this view, I show this particular one because it's depicted where the ATP is binding. So you can see the ATP binding to each subunit here. So as shown, 6 ATPs bound. So the structural diversity is quite tremendous, and here's just another example. So these are 3 different AAA plus ATPases of the ClipP system. So we're going to focus on ClipX, but it's not the only one. And so what we're looking at here is ClipX. We have another family member, ClipA, and here, ClipB, okay? And so what we see is subunit to subunit, whether it's X, A, or B, there's quite a bit of difference. Right? So ClipX is the most simple in terms of the architecture here for that. And so one thing people think about is in terms of the different activities that have been associated with these different family members, how is it that these different structural features play a role? Okay, here. So coming back to ClipX and the depiction we saw before, right? ClipX is an unfoldase, And what's really a key point here is that ATP hydrolysis by ClipX is going to power conformational changes that allow for mechanical unfolding of this protein that's condemned for degradation by ClipP. Okay, and that's what's going to also allow for translocation of the resulting unfolded protein into the degradation chamber. So the action of ClipX is allowing that protein to fit through this axial pore and be threaded into the chamber. Here. So with that, what are the questions we need to address in thinking about how this macromolecular machine works? One, how are substrates recognized? Right, so there's some certain group of proteins that are going to be degraded by this machinery. What is the mechanism? How is it that ATP-dependent conformational changes of ClpXP drive unfolding and translocation? And what is the substrate selectivity? So that's where we're going to move forward with. And so the first question we need to ask is, how are the substrates recognized by ClipX? Okay, here. Okay, and so what are possibilities? Okay, first, what we'll consider is a degradation tag. Okay, so when I draw these cartoons, I'm only going to show one of the two rings for ClipP. It's not that it's only one, this is just for simplicity. But imagine that here we have X. Here we have P. Okay, and we have some condemned protein, which I'll just draw as a circle. So the cell no longer wants this protein, it needs to go away. Okay, and we can imagine, as one possibility, is that a degradation tag can be attached to this polypeptide. Okay, and what we find is that there's a particular tag called SSRA tag that is used to tag proteins for degradation by ClpXP. So we can think of this tag as a zip code. If a polypeptide gets modified such that this tag is appended, it's going to end up going to this degradation machine such that it gets degraded. Okay, the tag is 11 amino acids. It's attached to the C terminus. Okay, and the sequence is A, N, D, E, N, Y, A, L, 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 A, A. Okay, and so what happens in this case as shown? Okay, we can imagine that this tag binds to the pore of ClipX directly, and the tag, when binding, enables translocation. So here, okay, and this pore has what are termed pore loops that are involved in tag binding. And in particular, there's a region GYVG, so 4 amino acid sequence that is thought to grip and drag the substrate. Okay, here. And of course, it's not gripping like we would, but there's some interaction there happening that allows that to occur. So you'll see there's a lot of kind of mechanical type cartoons and language used in describing these machines. Okay, so what is another possibility? Okay, so another possibility for how an ATPase could interact with a degradation chamber is that the protein substrate binds to an extra domain attached to the ATPase. Okay, and I point out this possibility is not for ClipX, but it's one to be aware of because it can occur. Okay, we saw some of those other ATPases are quite complicated. So in this case, imagine we have an ATPase. We have the degradation chamber, and this ATPase has some extra domain. Okay, that effectively can bind the condemned protein and help deliver it to the pore here. And just as a third possibility, and something that we'll see moving forward, is that there's involvement of an adapter protein. So in addition to the ATPase and the degradation chamber, there's an adapter protein that comes into play. So in this case, the protein adapter and this protein helps direct it to the pore. So the condemned protein to the ATPase. Okay, so for instance, here we have the ATPase. Here we have the degradation chamber. Okay, and maybe there's some additional protein that facilitates getting the condemned protein to the ATPase. Okay, and so something to keep in mind, and what we'll see with ClpXP, is that 1 and 3 are not mutually exclusive. Okay, and there's an adapter protein named SSPB that can help deliver SSRA-tagged polypeptides or proteins to the degradation chamber here. Okay. So we're going to think about these SSRA tags quite a bit. And something else to be aware of is just that these SSRA tags are not the only ways of tagging proteins for degradation. We're not going to talk about it in detail in this course, but you should be aware of something called the N-end rule. And this is really cool. So this rule basically states that a half-life of a protein is determined by its N-terminal residue here. And this can be called an N-degron. And these N-degrons are recognized by proteins such as ClipS and E. coli. And as a result, these proteins can get delivered to degradation machines. So for instance, in addition to ClpXP, there's an ATPase ClipA that can associate with ClipP and be involved in degradation of polypeptides via this N-end rule. Okay? And in terms of the rule, depending on the identity of this N-terminal amino acid, it may be stabilizing or destabilizing in terms of protein lifetime. If you're curious to know more about that, we can refer you to some literature. So here we have a cartoon looking at a native protein substrate that needs to be degraded. It's been modified with a tag. We have ClipX here. We have ClipP. Okay, here's the tag. And in addition, we can have this adapter protein SSPB. And the adapter ClipS. So let's think about this tag for a minute. And we need to think about this tag from the standpoint, one, of in vitro experiments, because we're going to begin to look at some experiments that were done to understand how this machine works. And we also need to think about this tag from the standpoint of the cell. So if we think about an in vitro experiment, where we want to study how ClpXP degrades some protein substrate, right? We can use this SSRA tag, and it's quite easy to attach 11 amino acids to some protein or polypeptide at the C-terminus, right? We can do that by protein expression. We can do that by chemical synthesis here. And so we're going to look at a number of experiments where this SSRA tag has been appended to certain model substrates moving forward. So what about in the cell? So when is this SSRA tag attached to a protein? So are all proteins that need to be degraded destined to ClpXP? Just intuitively, what do you think? Yes, you shaking heads, no, right? There's many, many proteases around. So what proteins are destined for degradation by ClipXP? That's what we're going to look at and how this tag is appended. And so effectively, this SSRA tag, say in E. coli, is used, one, because protein degradation needs to be tightly regulated, but two, it's used for dealing with proteins that have exhibited stalled translation. So this discussion is going to bring us back to the ribosome here. So we want to ask what proteins in the cell are tagged with SSRA. How is the tag attached to the chem protein as well here? This is just a cartoon showing an adapter protein helping direct this tag to the substrate here. So we're going to just move forward to this slide. This tag is specifically added to proteins that are experiencing stalled translation. So it's estimated that on the order of 0.5% of E. coli translations result in SSRA tagging. And so this is thought to be one largely of quality control, right? So you can imagine if the ribosome stalled, there could be buildup of polypeptide products that aren't wanted, and the translation machinery could get blocked, and we don't really want that to happen here. So here's our friend, the ribosome, and here's looking at the 50S ribosomal subunit, and we have a polypeptide emerging from the exit tunnel, so these should all be familiar at this stage. And so what happens when this ribosome is trying to synthesize a polypeptide and it just gets stuck? So this SSRA tag is attached to the C-terminus of proteins, and as we're going to see, this occurs co-translationally. And it's very, very interesting machinery. So what we see here is that there's a new player we haven't yet seen, and this is called SSRA, or tmRNA, for transfer messenger RNA. And it's involved in attachment of this SSRA tag to polypeptides that are having stalled biosynthesis on the ribosome. And so this player acts as both a tRNA and an mRNA, right? And we can take a look at the structures shown here. So here we have tRNA in the cloverleaf depiction, just an ala tRNA ala. And if we take a look here, what do we see? At this end, we have a region of the tmRNA that looks like a tRNA, right? Quite similar here to the 3'5' end. And then we have this additional region. And then if we look down in here, what do we see? We see a region that, with a little imagination, we can think looks like mRNA. And if we take a look at the various codons, what we see is that the SSRA tag is encoded there, along with a stop codon. So effectively, we have a tRNA lookalike, we have an mRNA lookalike that is encoding this SSRA tag. So what happens? There's a partner protein called SMPB, just to be aware of. And in complex with SMPB, it's actually EFTU that delivers this tmRNA to the ribosome here. So this is pretty interesting, just from the standpoint of what we know about EFTU. We don't have a typical anticodon here. So how does that happen? We're not going to go into details, but it's something, curiosity should beg those questions. So what happens? We can look at this cartoon overview here. And so the color coding within this is helpful in terms of keeping track of pieces. But here we start with our stalled ribosome. So the mRNA is bound. We see there's a peptidyl tRNA in the P site. The polypeptide has a number of amino acids, and the A site is empty. And for some reason, no new amino acyl tRNA is coming in. So the ribosome stalls, and as a result, this SSRA, or tmRNA, is recruited to this stalled ribosome. And so here we see the tRNA end in yellow with the alanine attached, and here we have that region that's mRNA-like encoding the tag. So this molecule, biomolecule, gets recruited, and what do we see? It enters the A site. So here we see the tRNA end in the A site, and we have the rest of the molecule here. Then what? There's formation of a new peptide bond, so we have peptidyl transfer. We see that alanine is here. Look, that looks quite a bit like the hybrid states we talked about, right, where we're seeing these ends shift into the E site not shown and the P site here. Okay, and then what happens? There's translocation and there's message switching. So the original mRNA gets kicked out, and what do we see? Now that mRNA-like region of the tmRNA is in the A site here. Then what happens? After replacement of the mRNA, translation can occur, which results in synthesis of the SSRA tag. So that's how this tag is attached to the C-terminus of the polypeptide. Okay? An elongation occurs until that stop codon in the tmRNA enters the A site. Okay? And then peptide release occurs here. So the result is a protein that has the SSRA tag attached to its C-terminus, and that protein will be directed to ClpXP. So pretty cool? Yeah, I think so. There. We don't ever leave the ribosome too much within these units. Here. Just to point out, this tag is universal in bacteria. So here's just a table of phylogenetic distribution. You're not responsible for these details. Yeah?
About the next slide. So is the tag attached after it's stalled, like is the original protein completed, or just the original mRNA removed and it attached the tag wherever it's stalled?
Yeah, so what does the cartoon suggest?
I mean, it feels like it's already on the C end here.
Well, the ribosome can stall at various points, right? So imagine you have a 100 amino acid polypeptide that needs to be synthesized, right? The ribosome could stall after amino acid 20 or 40 or 60, right? It's not that the whole polypeptide has been synthesized and then this gets put on, right? It may be some fragment there for that. So, yes. So in terms of this adapter protein, I just want to make a note in terms of the adapter. So these adapter proteins can help with regulating the substrate specificity of AAA+ ATPases. And effectively, depending on the system and depending on the adapter, it may enhance or it may inhibit degradation here. So it's a case-by-case basis. This SSPB, shown in the cartoon here, is a dimeric adapter for XP, and it promotes degradation of certain substrates. And effectively, it enhances recognition of this tag by the machinery such that the degradation rates are enhanced. So it's not that it's required, it's just helpful and accelerates the process. Here, so just here. So just an interesting observation regarding SSPB. It can be co-purified with ribosomes here. And in terms of its structure, it has some resemblance to known RNA binding proteins. And this resemblance has begged a question, does SSPB itself help with linking protein synthesis and protein degradation, right? So is it possible that SSPB could help promote binding of ClipX to polypeptides, before full release to the ribosome? That's something people have wondered about. And initially, this protein was classified as astringent starvation protein. That's where the name comes from. So if we just take a quick look at its structure here, what do we see? So here's SSPB, and then here we have structures from ribosomal proteins. And so in SSPB, ClipX binds on this side, and effectively here we look at the ribosomal proteins that bind RNA, and they have these RNA binding sites there. So there's some similarities in terms of the alpha helix, in terms of the beta sheets here. And also I'll just note in terms of SSPB and the SSRA tag. So if we take this tag, so this is our SSRA tag here. What's found is that ClipX recognition is on this end. Okay, and SSPB binding is on this end here. So in different points. So what this indicates here is that SSPB and ClipX can bind simultaneously. Okay, but this is small, so we can expect that there's some clash here for that. Okay. So where we're going to close is just looking at an overview as to how this machine works and the model that then, starting on Friday, we'll look at experiments that were designed and performed to inform this model. So if we look at this in one type of cartoon, what are the stages? So we can think of 3 as depicted here, where there's some sort of initial recognition. So the SSRA tag of this condemned protein binds to the axial pore of ClipX. And this process does not require ATP hydrolysis. So here we see a folded substrate. This degron is another word for one of these tags, SSRA tag. So we see there's recognition here. Then what happens? ClipX unfolds this substrate, so somehow it has to grip and pull and apply a force that unfolds the polypeptide and threads that unfolded polypeptide into the degradation chamber. So, you know, kind of this pulley system is shown here, this chopper type thing shown here. You can use your imagination in this unit for how to depict this machine. So we see that the polypeptide is being unfolded and threaded through ClipX into this chamber where it gets chopped up by the serine protease active sites here. Okay, so for unfolding and translocation, ATP is needed. ClipX is hydrolyzing ATP to allow this to occur. Okay, in the degradation chamber, this degradation part is independent of ATP, right? The serine protease doesn't need that here. So how can we kind of break this up further into a model that we can test? What I present here is the working model. And just note, the orientation is flipped here, so we have ClipX on the bottom and ClipP on top. So what happens here? We can look at this in terms of 5 steps, okay? And we can begin here with binding. Right? So this SSRA tagged protein needs to bind to ClipX, and that binding is associated with some dissociation constant, or KD, here. Right? What do we see? After binding, we have a second step, which is denaturation. So the polypeptide becomes unfolded. And that's defined by a rate constant for denaturation, as shown here. If we look next, we have translocation. So this polypeptide is moving through ClipX into the degradation chamber, right? And this is also associated with a rate constant, so rate constant for translocation. And both of these steps require the use of ATP. Once this polypeptide is in the chamber, we have step 4, which is degradation. And again, we have KDEG. This is fast. And then in this last step here, there's some release. So somehow these polypeptide fragments need to be released from the chamber.
Is ClipP still a dimer at this point?
Yes, yeah. So often the cartoons are drawn just showing one of the heptamers, but think of it as a dimer with these 2 back-to-back rings here for that. Right, so we have 5 steps here. Right, each one of these steps has a rate constant. And one question we want to ask with this is, what is the rate determining step? And the quick answer where we'll end today, and as indicated in this overview, is that degradation is fast relative to denaturation and translocation. Okay, and there should be an intuitive aspect to that. We heard about last time how proteases give these tremendous rate accelerations, right? And if you have an unfolded peptide, those sites where cleavage will happen are going to be exposed there. So what we're going to ask is it possible to make experiments, design experiments, where we can separate the denaturation process from the translocation process and analyze those, and in the process of doing so, ask, what is the ATP utilization for each step, and what is the role for ATP in this process? And so on Friday, we'll begin with discussing substrates, the design of substrates that have been used to examine this model in more detail. Is there one question, Max?
I was just wondering if the degradation step also removes post-translational modifications, or only things like that?
In the degradation step, that's going to depend. I mean, you can have different types of bonds with post-translational modifications, right? Right. So in the eukaryotic system, you have a post-translational modification to direct this condemned protein, and that machinery, so they're ubiquitins, and you get this polyubiquitin tail. So you saw ubiquitin in recitation number 1, and the eukaryotic proteasome has the ability to chop those ubiquitins off for recycling there in that. So that's one example.